Okay, thank you everyone for joining this workshop. And I actually took advantage of being home today to revamp this presentation. I've been giving this workshop for maybe 20 years now. So we have now some new slides, and I hope you'll enjoy the new materials. It's a bit more up to date than the previous presentation. So this is a guide, step-by-step guide to writing a scientific paper. But I will start by telling you that actually this workshop is not about writing. It's about putting together a paper, okay? And one point I want to make that I think is really central to this presentation is that writing skills are secondary. Frankly, if you're going to write scientific papers, the key part is not the writing. The writing will come afterwards. The key part is being able to really think a paper as a scientist, putting it together as a story. And that's basically the exercise I would spend most of my time talking about today, is how do you actually develop a scientific paper from your research, turning it into a published story that readers will enjoy reading and learning about your work. So one key message to start with is that writing skills are secondary. And another disclaimer perhaps is that I don't claim that the method I will present is the only way to write a paper, but I know that this method does work. It has worked for me. I've been applying it for many, many years, and it does work. And it is really a sort of protocol, step-by-step guide. So if your supervisors or the co-authors you work with have a different method, that's fine, that's great. But if you apply this method, it should work for you. Okay, let's watch some football. You guys like football? All right, so the reason I'm showing you this is actually from YouTube, and it's a video of the open goal misses. Okay, so you can see here some really beautiful actions. A lot of this is blurry. These are very old videos, but look at that. Oh, missed. So this is a little bit a metaphor that I'm using to tell you how important it is to publish, because I know that all of you are good scientists, are hardworking scientists. You, oh man, that one was bad. Huh? You spend a lot of time doing research, producing a lot of data, analyzing it, etc. But for some reason, the publishing part, which is the last step of the process of doing science, for some reason, that seems to be a limiting factor for many in their careers. And it seems to be something that, in my experience, a lot of especially early career scientists, are scared and worried about. So part of what I want to do today is really to demystify the process of publishing. It's nothing different from anything else you're doing. And really, you don't want to be in that situation of having all these wonderful moves like these football players here, and then be in front of an open goal and miss it. That would be absolutely terrible and devastating. And the reason for that is that our job as scientists is to produce knowledge, is to produce some science, to do some science, to produce some new information that is hopefully useful for society and useful for your colleagues. But our job doesn't stop there. Our job is also to communicate our science. It's not enough to just produce science, but also we have to communicate it. And to communicate it, publications are the main medium through which we share our science and share our findings with our colleagues. There are other means. You go to conferences, you present talks, you have posters and all that. But at the end of the day, the one medium that has developed as the main way through which we communicate science in a fairly organized fashion, in a robust fashion, is publications. So if you do a lot of research and don't publish, then it's really pity, because your work will have almost no impact. People will not really build on it, and it wouldn't have really achieved the function that it should have. When you get funded to do a research project, for example, the goal is not just to do research, the goal is also to publish it. So it's very important to publish. So I'm probably telling you things you know here, but I really want to emphasize this because for some reason I also find that many biologists spend a lot of time in the lab doing experiments, but don't spend enough time thinking about how to publish their work. And somehow that's often a process that's done kind of at the end of a project. You work on a project several years and at some point stop and think, oh, okay, let's publish it now. How will we publish it, etc. And I think that's not the best way to approach it. And that's the sort of things I will talk about today. So to back to this business of writing. So again, writing good papers is not about being a good writer. It's about describing and telling your research in a clear and rational fashion. It's explaining your work in a way that is understandable to your reader. And the best way really to tell something to someone is to tell it as a story. So in a way, great papers are really built as great stories. So writing papers, putting papers together is more than anything about storytelling. And that's the part I want to spend the most time about. So it's really about telling good story. What does it mean? It means also that it is about disconnecting yourself from the history of the research project. And this is something we'll talk about, and I think something that's very, very important. But then again, you see these three words that are highlighted: unknown, finding, and implications. So writing good papers is also about understanding the unknown that you have investigated, knowing the new finding and how it contributed to the field, and then understanding the implications of your findings. And I will spend again a lot of time talking about these three aspects of writing a paper, which are really central to developing a paper and developing your story. Okay, so what is the secret to publishing a lot of papers? Sure, all of you want to be very productive and very efficient and want to publish as many papers as you can from the research you produce. So the secret to that is an integrated research publication plan. What you want to do is you want to be developing storylines, developing outlines of your papers while you're still doing experiments. Why do you want to do that? You want to do that because it will allow you to find out what's missing in your story while you're still on the project and not after you're gone from the lab. It will allow you to know, okay, well, there's a piece missing in my story. I really should prioritize this experiment. And then that allows you to address it before you're actually before you actually left the lab. And you still have time and maybe funding for that project. Also, it gives you a paper concept. You have in your mind now an outline for your paper. You have a storyline for your paper. And this is very important for your intellectual exploration of that topic. It will allow you to think about your research in a different way because you have a concept about the story you want to tell about your research. And that might actually open up a different perspective to your work than you would have otherwise. And also, it really guides you in terms of talking to your colleagues, reading the literature, etc. You might present your work at the conference, and somebody might come to your poster or listen to your talk and ask you a question in a certain way that might give you an idea about your paper. And because you have that paper concept already in place, it will be easier for you to integrate that question, that different angle to your paper. Okay, so that's kind of my general thoughts. And now I'm going to move to the essentially the protocol of writing papers. And this is really truly step-by-step guide in 10 steps on how to write a paper. And as you see from that cartoon I put there, and as I'm sure you all know, any complex project is best done by breaking it down into individual steps and focusing on this one step at a time. Okay. So this is important in your research. It's important in a lot of things you do in life. And this is also important for writing papers. So what I'm offering you here is really a 10-step protocol to basically go from your research from a position where you have obtained some promising results in your work, that you start thinking, okay, there's a paper really building up here. I have some exciting findings. My experiments are starting to come together, and I may have a paper here, to the process of actually finishing it and publishing. So that's what I will do next, is I will go through this protocol essentially step by step, and we'll discuss each one of these. But before I do that, are there any questions? Does anyone want to chip in? Okay. All right, so let's start with the first step, which is creating a folder. This may seem a little bit simple and obvious, but actually it is important in this project. I noticed that many people don't actually create folders related to their papers. They have folders with their data, folders with all sorts of work they're doing, or maybe folders with the literature, but they don't create a folder with of the paper. And these days, you know, if you're using Dropbox or using other ways to share files with your colleagues, then create a folder as soon as you have an idea about your paper, and share it with your co-authors and your colleagues. So that way everybody can chip in and add information to that folder, etc. So what this folder becomes, it is fairly early in the project, right? So this is at the point where you start seeing that, okay, I have a story here. I may have a paper. I have some ideas about the story to build the paper around. Okay, let's start discussing this. So you're still doing research. The project is by no means completed, but that folder becomes like a place where you can collect information related to the project, to the paper, right? Literature, ideas, etc. You might also be inspired to write something and then write it and put in that folder. So I personally use the phone a lot to write stuff, and I'm often inspired in the morning, for example, maybe because I dream about stuff. And then you wake up in the morning and you have these ideas and it's all very clear in your mind. Well, write a few sentences, that's okay, and then put them in that folder. That might become part of your discussion later on, or part of your interpretation of the paper, or part of your introduction, etc. So this creating that folder is actually a step that allows you to get organized towards the process of publishing a paper. And of course, you may have more than one folder, because you might be working on multiple papers at the same time. Certainly, as you get more advanced in your career, you'll be in that situation. Then it's important to actually operate through that process of having your papers organized and your ideas organized in a clear fashion. Okay, so now we created the folder and we shared it with our co-authors, and we start brainstorming about a storyline. So what is a storyline? Okay, so the storyline is really the concept of the paper, but told as a story. And the point that's important here more than anything is the storytelling. And like every good story, it has a beginning, it has some sort of question or some sort of puzzle. There's something that is found or discovered, or something that addresses that puzzle. And then there is a conclusion and some sort of implication maybe of that conclusion. So these are the key sentences of your storyline. And this is very important to start working on that early on in the project. And also it's very important to keep going back and rehashing them, discussing them with your colleague, discussing them with even people who are not involved in the project, because that's just a few sentences. It's very easy to actually kind of stress test them and see if people like them or not, to see if they actually fit or not, etc. So what you want really here is write maybe a sentence of background. And then the two most important sentences, and this is when we do this workshop and we do the exercise and people bring drafts of their papers, these are the two sentences I want them to focus on. The ones there in red and in blue. It's the unknown and the finding. And when I say write a sentence, I really mean it. Just one single sentence for the unknown and one single sentence for the finding. It's very important not to have a whole list of findings. Even if you have multiple findings from your project, you have to try to work hard to distill that into a single sentence. And that's not easy, but that's very important. That's very, very important for you to give your paper a focus. And then, of course, you have some resource statements, and then we'll talk later about implications, conclusions, and implications, but that comes afterwards. But let me go back to this unknown and finding, because this is really important. So the unknown is essentially a problem or essentially a question that's still open in the field. And that can be quite broad. It's okay to be broad. Don't feel like you have to be extremely focused. And also, when you write the finding, it's important to realize that your finding is not going to completely answer that question. It's not going to completely answer the unknown, but it will provide one piece of the puzzle. So if you're presenting a big question and you're contributing partly to that question, that's perfectly fine. It's better than the other way. It's better than having a very complicated list of findings and a very fuzzy or very narrow unknown or question. Okay, so when you do the outline and the storyline, usually along that process, at that point you already have some data and have generated a bunch of interesting, promising data. So while you work on that storyline, this is also the time where you start making a list of figures and tables. Okay, so you already have some data sets, some data displays you generated because you've done experiments that are promising. So start drafting a concept for the figures and the tables. Make a list of these figures and tables and start thinking about a list of results sections. So start thinking about doing some outlining of your paper and make sure that your results sections match the figures. So start thinking of like, okay, this is my story. So what should be the list of results that I have associated with that story and associated with the figures? And also an exercise that you can do here is really give each figure and results section a title. But that title should not be a description of an experiment. You shouldn't say, for example, gene expression analysis of gene X. You should say, you should make an assertive title. You should say gene X is expressed during infection of this plant by this pathogen, okay? So that's very important too, because that will help you sharpen again your views about what are the conclusions coming out of your work, what are the findings, and rather than sort of describing experiments, it forces you to really think about what your experiment really has generated in terms of a finding. Okay, and so now you're starting to sort of have an outline that's quite serious because you have a storyline and you have a list of results sections and a list of figures and tables associated with those results. Now, when you have that, that's not really something that is done. That's something that's quite dynamic, especially in the early stages of a project. It's something that you will really keep working on. You might go back and say, oh, you know what, my story would be better if I tell it this way. If I start with this particular figure, with this particular data, and then move the other stuff towards the end, for example. Okay, so this is something that is normal and perfectly encouraged to basically continuously brainstorm and going back and forth between the storyline and the figures. It's an iterative process, and also I'll talk about that in a second. This is happening while you're still doing research, so you may have new results and new findings that don't totally match that storyline, and you may have to go back and revise it. So a pro tip here, one of these hashtag pro tips I would be giving you, pro tip is don't number the figures. This is a very common beginner mistake. Don't number the figures, because if you number the figures, chances are that those numbers will change, and that's going to be a complete mess. Figure 1 might become figure 5, and so on. So if you start doing that from the beginning, it will kind of box you into a hole where you think that's the order of my figures, it's never going to change. Whereas that's something that should be very flexible until the end, really, because you may have an idea of adding an extra experiment or something that will improve the storyline. So pro tip, don't number your figures. Give them names like figure, gene expression, gene X, something like that. Okay, and now a warning. So I told you that this is done while you're doing research. You'll be developing storylines that are in a way intellectual exercises to some degree, because you don't have all the data yet. And it might guide you and push you in doing some experiments, because you do need those experiments to complete that story. But outlining the paper while doing research should not, under any means, imply that you become wedded to that storyline. As we say, the tail shouldn't wag the dog here, right? So the data should be guiding the paper, not the other way. Okay, so please really think very hard about this. It's not an exercise where you make a storyline and then you start delivering data that fit that storyline. That's not how it should be. The storyline should be totally revised as contradictory data emerges. It's an intellectual exercise that you will be doing in many iterative ways. And I can tell you that this hard disk in front of me right here is full of many hopeful storylines that would have been nice if they were true, but they did not pan out when we did the experiments. Okay, so the storyline. I mean, the storyline is very important. It's really important to think about it and spend time thinking how to tell a story, right? So how would you tell the story, okay? Well, you could tell it the boring way, as I like to say. The boring way is generally the historical way. Telling this experiment exactly how they were performed. And this actually rarely happens. Most papers do not follow the historical orders. And this is what led Peter Medauer, Nobel Prize winning immunologist, British of Lebanese descent, really wonderful writer and wonderful scientist. He actually wrote this essay titled, "Is a Scientific Paper a Fraud?" And he's absolutely right. What he means by that, by the scientific paper being a fraud, is we don't write scientific papers by telling how we actually did the experiments. Frankly, many of them will be quite entertaining then, because so many discoveries are accidental, right? So you say, oh, well, I was looking for whether gene X is involved in this process. I made a mutant, and then turned out to be completely different from what I expected. And I get this. So it's usually not written like that. So one of the hardest things to do when you're putting together a storyline is to actually disconnect yourself from the historical order, from the way the experiments were done, step back and think about your data in maybe through a different lens, from a different angle, and try to think if you can actually make a better story or more interesting story than the one you actually had when you started your project. Because most of the time, the outcome is not as expected. And when we write proposals or write research proposals or DTP studentships or any of the sort, when the experiments are actually done, when the project develops, it's rarely as expected. So the one thing you should not be doing is just basically put your data in the historical order and say, that's it, that's my paper. You could do that, but that's not really necessarily the most exciting way to do it. Well, another way to tell your story in maybe less exciting way is to basically start your story with some generalities. Like I put here an example, proteases are important in plants, therefore we studied them and maybe wrote a paper about them. Or in my field, for example, in plant pathology, many papers start by saying Phytophthora infestans is an important pathogen of potato, and therefore we study Phytophthora infestans and looked at whatever this gene does, or etc. So this is fine. I mean, this is nothing wrong with that, and many papers actually start like that. But if you think about it, you're already narrowing the frame of your paper. You're actually framing your paper in a way that is interesting to only a few experts in that field. For example, somebody who does work on Phytophthora infestans may not be interested in your paper. Somebody who doesn't work on proteases will not be interested in your paper if it starts and framed around a pathogen, an organism, or a class of proteins. So an exercise that is worth trying is to step back and try to frame your paper in a way that is interesting to people outside your field. That's a challenge, and that's an exercise. And that's actually the exercise we do in the workshop. When you write your unknown and your finding, especially your unknown, try to think about how to frame it in a way that is targeting a broader audience than the one you would normally target or than your immediate field. For example, you may have a pathogen gene that is interesting to people who study animal pathogens. So why should you frame your paper around plant pathogens? It might go beyond that, and so on. Maybe the process you found in this protease is relevant to other proteins that are not proteases. So it's worth trying. We all agree that not every paper would be actually of general interest. That's fine. It's still very valuable research. You would still have contributed to science and to knowledge. And the point here is not to turn every paper into a paper of general interest, but it's worth going through that exercise, because sometimes you will actually realize that, wait, I never thought about it that way, but yes, indeed, this paper could be interesting to people outside my field, and this is why. Okay, so how to tell your story in a more inspired way? This is probably the most important slide of the presentation, right? So if you want to pay attention, this is the time to do it, because this is really the key, is those two sentences, as I told you, two sentences you want to write. The first one is defining the unknown, okay? So here you're going to write in one sentence the biological question or the problem, okay? And again, this can be framed in a more broader or general way. And then after that, you're going to write another sentence, which is explaining your research finding and how your research finding has contributed to this new information question problem that you already stated. This is an exercise where you can go back and forth between these two sentences, try to sort of make them match and so on. And you may adjust your finding based on that unknown or vice versa. This is a very important intellectual exercise. And also, this is an exercise where you might actually write 2, 3, 4 versions of these sentences and see which ones are actually more exciting or more interesting. You may share them with people outside your immediate collaborator network and ask them what they think, etc. But this is the first and key step to putting together a great paper, is writing these two sentences, the unknown and the finding. Why did you do it? What did you find? Simple. And honestly, if this is not crystal clear in your mind, if you don't have a sharp view about those two sentences, writing the paper is going to be a struggle. It's not going to be fun and easy to write that paper. But when you have those two sentences, when you realize, haha, this is really clicking together now, then it becomes really easy to write a paper, because you have such a focus. You have such a sharp focus on what this paper is about that it becomes a lot easier to write. Okay? And this exercise is not easy. This slide is about reminding me to tell you that this exercise of defining these two sentences is not easy at all, because you're so specialized in your project. You're so deep into that project, working on it every day, doing all these experiments, worrying about the day-to-day details, that it's very hard to think outside the box, to step back, many, many steps back, and think about your research and your findings in a different, through a different angle. It's really hard to do that. And this is, I think, an exercise that you have to really work on. I found it useful to put together the storyline, put together the figures, and have a meeting and invite a couple of people who are not actually involved in the project, because they will have a completely different, fresh angle looking at your story. And whereas people involved in the project, they're already so boxed into thinking about it in a certain way that sometimes it's really hard for them to step back and think about it differently. So this is an exercise that's worth doing, stepping back and thinking about your research and your story through a different angle. I'm going to tell you a story here from one of the workshops, because I thought that was really beautiful. We had a student, she was doing a suppressor screen in Arabidopsis, genetic suppressor screen, and she had the mutant that did suppress the mutation that she started with. But that really didn't make a lot of sense. The phenotype was not so clear, etc. So she was really struggling with that story of explaining this second gene as a suppressor of the first one. But then she crossed it out. So she had the second gene, the suppressor, mutated in the wild type background without that original mutation. And then she had a really interesting phenotype out of that. A really interesting phenotype. And then she got intrigued by that. She read a lot about it. So she knew a lot about that second gene and the phenotype associated with it. She thought was really, really cool, etc., and that it was adding something to that field of research. But when she was thinking about her paper, she kept thinking about, as this is a suppressor screen, and she kept bringing it back to that first gene. But actually, if she steps back and think about, I have a second gene, I have a mutation, it's an interesting mutation. Who cares about the suppressor screen? Who cares about the first gene? Then it was a much sharper story. And when we went through the workshop and somebody suggested that to her, her eyes really lit up, and she said, yeah, of course, that makes a lot of sense. Yes, indeed, this mutation explains a lot of things, etc. And then all of a sudden the story came together, but it really had no connection whatsoever to how that project historically developed through that suppressor screen. And it wasn't actually what her supervisor wanted to find also, because they did the suppressor screen with another intention. But actually she did discover something accidentally that was really interesting and worthy of publishing. Probably would have made a much better paper than the original intention of writing it as a suppressor screen. So the point is, always talk to people outside your field. If you think you have some interesting story that is kind of boxed into a certain narrative that doesn't always come together as you want. Okay, an example of a storyline. All right, so this is from some of our work on pathogen effectors that target autophagy. Let's quickly go through this. So plants use autophagy to safeguard against disease, background sentence. However, how plant pathogens interfere with autophagy is unknown. Here we show that there is a pathogen effector that binds to an autophagy protein and stimulates autophagosome formation. And then some results. PEXRD54 depletes this receptor to do that stuff, blah, blah, blah. And then this shows that the plant pathogen effector has evolved to antagonize host autophagy. So we're now answering that with this conclusion, we're answering that unknown. And then at the end, there's an implication, which is a more general sentence. This would highlight the intricate changes in endomembrane compartment formation that take place during plant pathogen interactions. So that's an outline, and now I'm breaking it down into these sections, and you can now clearly see what these simple sentences, how powerful they are, because each sentence really very sharply communicates. First sentence being a brief sentence of background. The second sentence is the unknown. How plant pathogens interfere with autophagy is unknown. And we don't completely answer that question, of course, but we do contribute some knowledge that addresses that question. And then we have our finding in one sentence again. We show that this effector binds a host autophagy protein to stimulate autophagy. And then we have a bunch of results. And then at the end, that last sentence is very interesting. I want to think about that sentence for a moment. This work highlights the intricate changes in endomembrane compartment formation that take place during plant microbe interactions. This is a very generous sentence, and it really goes beyond what we found. And that's the implications. And I want you to really appreciate the difference between a finding and an implication. That difference also should be very crystal clear in your mind when you write your paper. Don't mix those two together. Basically, the finding is your result. It's very specific. We show that an effector here binds an autophagy protein and stimulates autophagy. But that finding always has an implication. It always has some sort of basically consequence on how we understand a certain field, and it allows you also to speculate a little bit more about what your finding means. And that's what the implications really means. This work highlights the changes in endomembrane compartment formation. Yes, it does, but we actually don't have a lot of experiments showing that we have changes in endomembrane compartment formation, etc. So it is really something that goes beyond what you actually did in that project. So again, to break down the storyline in 2 sentences, these are the different sections, and again, especially the unknown, finding, and implication, those should be single sentences. Now, the brief background and the results and so on can be multiple sentences, but that's basically your storyline. Okay, is it clear for everyone? Does anyone have questions or comment? No? Okay. All right. Okay, so one thing to keep in mind when you're working on your storyline, and really that emphasis on having single sentences and single statements, is really to push you to think that less is more. The best stories are the ones that are really focused without being bloated. Sadly, I have to say that many papers in biology now that are really bloated. They're just like collections of data. There's so much data, and it's just so many experiments. It's very hard to follow. And sometimes you're really like wondering why are they adding this experiment? Is this really properly done? Is this properly controlled? Is this replicable? So think about it, less is more. The best papers, the best stories are the ones that are focused. Less is more, as true in art, it's true in science. And I also want to emphasize this. This commentary was about triangulation. Triangulation is really something really important I hope many of you appreciate. That the best science is not science that is just simply replicable, that you can replicate and so on. It's science where you have multiple experiments, multiple independent experiments pointing in the same direction, pointing to the same conclusion. So the best papers are not the ones that are bloated with 10 different figures with 10 different experiments. The best papers are the ones where they have 3 or 4 figures all pointing to the same conclusions, maybe looking at the data from a different angle. Let's say you have interactions between 2 proteins through an assay, like yeast to hybrid assays. Then maybe you have now done additional experiments like implant association or in vivo association between these 2 proteins. Maybe have some in vitro data showing that these 2 proteins bind each other, or maybe have some cell biology evidence also pointing that these proteins are localized in the same compartment. So it's very important to realize that the best papers are not bloated with 10 sort of half-baked experiments that are all going in different directions and making the story really complicated. The best papers are the ones where there is a single focus, and that focus is very well supported, not by simply repeated experiments, but also through, as this commentary says, through having independent line of evidence all pointing to the same direction. That's the most solid science. That's a science that is more likely to stand the test of time, which is really what you want to achieve with your research and with your papers. Okay, so now we finish the storyline. We have our list of figures, our list of tables. You're now on a mission. And your mission now is to finalize the figures. And this can take a while because it's not easy to generate publication quality figures. This is also why you should integrate the process of doing research with developing the paper, developing the storyline, and outlining the paper. Because you might realize that, well, I should really add this control in this figure. I should really make sure that I have the same proteins or the same basically treatments in my different data sets, in my different figures and so on. You might actually have to repeat experiments that you've done many times because you're missing something that fits in the paper and so on. So really your mission, which I hope would not be impossible, just like Ethan Hunt, you are going to go and generate final versions of your figures and tables. And this is where you really have to make sure you cover the basics. So this is, a lot of this is technical. You have to make sure you have the proper replicates, you've done the quantifications and so on. And this is also something where you have to spend time learning, generating high quality figures and displays. So if you haven't done it yet, learn R, learn how to display your data using tools like ggplot. Cosmetics does matter. You may want to talk to your colleagues about even colors, are you using the correct color combination and so on. And learn from the best. When you read papers and you find figures that you really like and you find are really clear and easy to understand, then get inspired from them. The point is that this is really an important exercise that needs to be considered and also requires some training and some learning on your part to learn how to make great figures. This is an example of that. In these days, we really are moving away from histograms, for example, to display data. So learning how to display data is something that's really important, should be part of your training. Realizing that, well, histograms, is that really the best way to convey data to your readers and to convince your readers and reviewers and so on about your data when you have much more powerful ways to displaying data, especially these days with tools like ggplot and so on. And this paper, which is really it's not so old, it's only 5 years ago, but it's really talking about a new paradigm for data presentation. I'm still shocked at how many papers have just regular histograms where they could show you these scatter plots, which are much more informative. And what this figure shows that that histogram basically would match all these other 4 different data distributions. They will all look the same in that histogram, even though they are, as you can tell, there are really different outputs for these experiments, very different outputs from what the histogram may convey to reader. So learn this. Take the time to figure out basically the best way to display your data. Talk to your colleagues. There are some trainings that are available and so on that are worth taking time to do. All right, so now we have your figures. You have your figures and tables ready. This is when you start writing. So don't start writing before you have your final figures and tables. Don't start writing the results, I mean. And the results really are the next thing you would write normally in this process. The reason why you don't start writing until you have your final figures and tables, because really when you have those final figures, first of all, your storyline will really be now cemented. You know exactly what your storyline is about. And also because your results, they should describe what's in the figures and all the data you provide and not something else. If you start writing too early, then your figures and your data set might be outdated. And then the worst thing you want is really having your results not matching very well with your data displays. And that would really irritate reviewers and editors. And this is also when you write, there is a tendency to make things sort of streamlined, but I think it's okay, and it's important for you to realize that the point of writing your results or describing your experiment is not to turn everything into binary sort of, oh yeah, we observed an effect and that's it, or we didn't observe any effect. Is if the data is quantitative, then do mention that. Do mention that it is quantitative. Provide that data. Maybe you provide in supplementary material if it doesn't fit in the main figure. This is also the place where you really should report about replication statistics that might be in the figure legend, but it might be worth emphasizing if it's relevant to the results. Okay, let's go through results paragraph. Results are technical, so the text I chose here is technical, and that's really how the results are. They're usually the most sort of tedious part to read because they're full of experimental details and information, so it can be challenged to write a good results section. So this is an example of a paragraph. So let me take you through the story very quickly. Again, it's a bit complex, but that's fine. That's how it usually is. So the ZAR1 model postulates that the resistosome translocates into the plasma membrane through this alpha helix, which matches this motif we identified in this paper. To investigate the intracellular dynamics of this motif, we expressed and analyzed the subcellular distribution of an NRC YFP construct in Nicotiana benthamiana leaves using this method agroinfiltration. This construct produced fluorescence signal in punctate structures throughout the cell and also some weak nucleocytoplasmic signal. The pattern was different from free YFP, which usually was nucleocytoplasmic. To further study this construct, we examined puncta formation by a mutant of YFP, which is deficient in self-assembly, in contrast to the original construct, that NRC 129 YFP. This YFP mutant rarely formed puncta. We conclude that YFP oligomerization is required for the NRC YFP to form puncta in benthamiana. So this is, again, breaking down this results section into these different subsections. And again, this is a very useful guide for you when you're writing your results. So the first sentence is optional. It's a background sentence, introductory sentence. it's usually 1-2 sentences, not more. If you find yourself writing whole paragraph here, then really that paragraph belongs to the introduction. But sometimes in the flow of the story, you may want to add some background sentence to really help the reader sort of refresh their mind about what is going on here. The second sentence, the one in red, it's an easy one. When you write it, you always start by saying, to do this, we did that. Okay, so here to investigate the intracellular dynamics of this motif, we expressed and analyzed this protein in pentamiana leaves using this particular method. And then the text in blue are the results. So these are your results. And usually what you do here, and again, that's a useful tip, is start by telling your most important result. And then bring up any additional experiments you've done or any controls. So you can see here that that first sentence does not, that first sentence here only gives the main results, not talking about the control, for example. And then the second sentence is the control, the patterns, unlike the free wife, right? And then there is additional data that is shown afterwards. And then your final sentence is your conclusion. This is usually one sentence, and it basically summarizes the finding, essentially, of your results section. And often this sentence is very similar to the title of that results section, which is the assertive statement about the main finding of that results section. Okay, so this is a very straightforward process where now you have essentially a guide to drafting your results section. Again, as I just went through it, background, to do this, we did that, that's a rationale, then some results text, and then a conclusion, which is the last sentence. Now, this is how you start. So if you start with this, it will make life a lot easier because you can start drafting it like that. Now, when you have the full paper together and when you're editing it, you might actually start changing this. It's probably a little boring if every result section starts with to do this, we did that. But that's okay. Start with this process as a first draft, because this allows you to basically generate that draft fairly quickly. And then when you revise and edit, you can start polishing. For example, to do this, we did that, may become like to test this hypothesis. Maybe that background sentence becomes a hypothesis, like we hypothesize that this protein accumulates in the nucleus. And then to test this, we did this, or maybe a finding from earlier in the paper. And then you could start by saying, this result prompted us to investigate whatever, right? But again, the structure of having a background sentence, the rationale for why you did this, the results, most important results first, controls and so on afterwards, and then a conclusion, which is generally the title of that results section, is a very straightforward process to get you going and really generate at least the first draft of that paper fairly quickly. Okay, I also mentioned about reporting data accurately, and this is important because you want to be able to follow up. I mean, once you publish your paper, you want to do more research on this. Other people will try to replicate your work. Your students, postdocs, other scientists will follow you on this project. They will use your material and replicate the work. So it's important when you have quantitative data to display it in a quantitative manner. This is actually the experiment matching that paragraph I just showed you, where we were talking and saying that this construct makes puncta and the others don't. But as you can see here, this is not a binary result. This is a quantitative result. And you can see that the number of puncta vary from what maybe 3 or 4 to 16 or so. And in the controls, you can have up to maybe 2 puncta, maybe 3. It's important to show these results quantitatively because they are quantitative. Nothing prevents you, doesn't make your data any worse. And then when people repeat this, imagine somebody repeats this experiment, they have lower expression, they may not actually see the puncta, they might be on the lower end, they will say, oh, your work is not reproducible. So if you report it accurately and report actually the variation you observed, then even when people repeat it, they can interpret their repeats in a more accurate fashion than if you say just, okay, this guy makes puncta, the other one doesn't, end of story. Okay, So this is an important also message, is to always try to report your data in as it is really, and not try to turn it into binary type finding. Okay, so now we're kind of almost there. Like if you think about it, when you're at this stage, so let me go back. When you're at this stage where you have finalized the figure, and it should go really fast because now you have the final figures. I mean, this is hard work. Okay, finalizing the figures can take a long time because that might also require repeating experiments, doing additional replicates, and so on. Maybe even revisiting experiments that have already been done. But once you have those figures completed and you start writing, this shouldn't be very difficult, because if you think about it, it shouldn't take you more than a day to write one result section. This is like writing an abstract. Like, I'm sure all of you can write an abstract in one day, right? When you go into conferences and you write abstracts, it's what, 200-300 words. That shouldn't take very long. This is really what this is. This is about a few hundred words. So if you think about it, it shouldn't take you that long to, if you have a paper with 7 figures, that should be 7 working days, right? So that shouldn't be that long to write actually all the results, right? So think about like that. Think about like, okay, I'm gonna give myself an objective. I have now all my figures. I'm gonna write one result section a day. Once you have the figures and you have the titles of those figures, which is already actually one sentence in that paragraph, then you're basically almost there. Okay. So the point is, again, if you break down your the process of writing the papers into these smaller pieces and smaller exercises, then it becomes something that you can really, so you can start seeing the light at the end of the tunnel. You can really see how you can progress with this project in a steady way. Okay, so now we wrote the results, and now we get to the introduction. So at this point, you can write the introduction, the discussion. This doesn't have to be completely linear. This is something you may have already written some of it in your earlier reports and so on. So it's not really critical whether you do the introduction before or after, but certainly the final version of the introduction should be done after you completed your results and finalized your results. Okay, what's in the introduction? The introduction is quite straightforward, really. You want to basically say first, why did this work? Which again, if you've gone through the exercise of writing that unknown sentence, that's already there, because you're going to tell the word what the unknown is. So that's quite straightforward. And also, this is where you provide background information to readers so they can understand the results. It might be using, I don't know, biomaterial or proteins or mutants in those results that require some explanation. So this is where you explain them. Usually, this is also where you explain your experimental system. Might actually give some background on previous work that's been done here. And then you also finish the introduction with a brief summary of the findings. So this is the protocol to writing the introduction. Okay, so first of all, structure. I think in any writing, and this will come up again, this came up earlier in the storyline, in the results, but every time you write, you really should first structure and outline what you're planning to write. So what you want to do here is you want to have 4 to 6 paragraphs of 200-300 words each, right? Most journals, the format would be an introduction of what, 1,000 to 1,500 words roughly. Sometimes they're shorter, sometimes it can be a little longer, but it's about that. It's about 4 to 6 paragraphs of 200-300 words each. So what you can do here is really you are going to assign topics to each of these paragraphs, make an outline, make sure that it flows logically. But the first and last paragraphs are easy, because they're always the same ones, in my view. The first one is where you take that sentence you wrote about the biological question, the unknown, the problem, and you turn that into a paragraph. So now you're starting the paper by stating what the problem is, what the unknown is, okay? And that sentence now, you develop it into a paragraph where you go a little bit beyond that one sentence. But since you have already written that sentence, it's actually fairly easy to go from that one sentence to a paragraph, because it's quite clear. For example, if you're talking about, we don't know how pathogens interfere with autophagy, then that's a very sharp focus of that one paragraph, and it's very easy to do a literature review and say, well, actually, we don't know much, for example, right? Or this is what's been known, or it's been known in animal pathogens, but not in plant pathogens, stuff like that. Okay, so first paragraph is about the unknown. The last paragraph is a paragraph that usually starts with the sentence, in this study, we did this. And this is where you generally kind of briefly give an outline of your findings, okay? So this is where you summarize your findings. So you say, in this study, we did this and we found that, okay? So what you did and what you found. So if you think about it's 4 to 6 paragraphs, we already have 2 paragraphs decided. So you really have to write essentially or be creative about 2 to 4 paragraphs, that's it. So so that's it. That's really the introduction. It's not more complicated than that. Just a few paragraphs, and again, each paragraph is 200 to 300 words each. So if you write 1 paragraph a day, then in 5 days you'd be done. You'd have written your introduction. Another hashtag pro tip is when you finish outlining your introduction, go back and look at your results, look at them section by section, look at your figures. You might find, for example, that you use another protein as a control in your experiments, and that there's some biomaterial or some topic that pops up again and again, and maybe you haven't covered that in your introduction. So maybe then it's worth adding a sentence or 2 in the introduction about that that bit that is maybe a little bit tangential to your study, but is present in your experiment. So that's useful exercise to do to go through your results and see whether everything was covered in the intro. Okay, now the discussion. So discussion, I love the discussion. Okay, so it's always my favorite thing to write. And the reason why I like the discussion is because this is the part of the paper where you have the most freedom to actually really go and intellectually explore what you've done here and what is coming next. This is actually your opportunity to establish intellectual leadership in your field by pointing to the future, really, by pointing to the possibilities and future directions that are raised by your findings. Think about it this way. So you done all this research, you made these new discoveries, and you're writing a paper about them. Because your discoveries are new, you're in a position to tell the world about what's going to come next. Okay? So this is what I mean by the implications. So it's, again, very important for you to have very clear view of what are your findings and what are the implications of your finding, and really separate these always in separate sentences, in sort of separate parts of the paper. It's very important to appreciate the difference between a finding, which is a very specific result from your experiment, and the implications of your finding. And the discussion is really where you are going to discuss the implications of your finding. And of course, this is where you discuss also how your findings relate to previous knowledge, to the state of the art, and then you found some things here. And I want to highlight this old paper with perhaps the most famous sentence in biology. It has not escaped our notice that the specific pairing of the double helix immediately suggests possible copying mechanism for the genetic material. So this is the famous Watson and Crick. We know the story that sadly there actually wasn't their data, it was Rosalind Franklin's data. So it's not like I even had data here. But that sentence is an implication sentence. They haven't done a single experiment in this paper to show that the double helix has anything to do with a copying mechanism of the genetic material. This is not a finding. Their finding was simply that they propose a structure for DNA, which is a double helix, based on Rosalind Franklin's data. But this sentence, which is really why this paper is so famous and their discovery was so important, is actually an implication. It has not escaped our notice that this is a potential copying mechanism. And this is really what you should think about with your data. When you discover something, you want to be the one who tells the world what it means, what are the implications of those findings. You don't want someone else to come and tell you, oh, by the way, you know, it has escaped your notice that this could mean that, right? So, so this is, again, the discussion is the place to do that. The discussion is where you have some leeway in terms of exploring the what your data really means, and further speculating about how that would affect your field and how that would affect other scientists. Okay, back to the protocol style outline here. So first of all, structure again, like always, assign topics to each paragraph, make an outline, and just like the introduction, this is really for most journals, it's 1,000 to 1,500 words, 4 to 6 paragraphs of 200-300 words each, something like that. So it's roughly again one paragraph a day, and in a week you're done with the discussion, right? First paragraph is generally a summary of the finding, so that's pretty easy to write. You basically go back and recap your findings in a very general way, maybe through a slightly different angle than you did before. The discussion is also where you relate your work to prior literature. Sometimes it confirms prior literature, sometimes it doesn't, and this is where you discuss that. You can say, okay, we observed that this mutant has this phenotype, but in the paper from 2 years ago, they didn't report that phenotype. Maybe we had different background, maybe we grew the plant in different conditions, etc., etc. So this is where you discuss all those things. And also, this is where you try to go beyond what has been previously published. Again, talking about implications and what your results mean for the future of that topic you're studying. Another hashtag pro tip: when you get stuck, and discussions are generally described as the hardest part to write because they are actually requiring a lot of intellectual input, and also it requires a very, very, very good knowledge of the literature. You cannot discuss how your work relates to other work if you're not familiar with the literature. So that's very important to obviously know the literature, and that's why stuff to write discussions sometimes is slow, because it often means that you have to go back and read many other papers to really refresh your ideas about what the state of the art is in your field and so on. But if you get stuck and you don't know how to start a paragraph, start with a question, okay? So you have some observations like, why does protein X alter the expression of plant gene Y? Okay, so that's a question. That would be your first sentence in that paragraph, and then spend the paragraph answering that. Or why does protein X alter the expression of gene Y in potato, but not in Arabidopsis? Something like that, right? And now you can sort of spend that paragraph discussing that, answering that question, if you like. That's a very good tip to help sort of break writer block when you're stuck with the discussion. Okay, so we're done. Huh? We're basically done. We wrote the whole paper now, and now we just have to do the title and abstract. And the title is important. The abstract may be less so in my view, but we'll talk about that in a second. Why is the title important? The title is very important because this is your billboard. Think about it as your billboard, right? That title, think about how many people will read the title compared to the number of people who read the paper. I don't know. I have a guesstimate, I guess. I would say for every 100 people who would end up reading the title of your papers, maybe one of them will read the paper. Maybe it's 100 to 1. I don't think it's much better than that. Maybe for 100 people who read the title, maybe 10 of them will read the abstract, and maybe one will read actually the paper. I mean, the titles will be in, in your CV. There will be in all sorts of stuff. They will be online in tables of contents. When people do searches, they'll find the title. It's just going to be everywhere. It's going to be in your grant proposals. When you write fellowship or grant proposals, you will list your publications. The title will be there, not the abstract, not the paper. So spend time thinking about the title, and spend time giving the reader something useful and interesting to them. And by doing that, you do that by actually writing ASTs, assertive statement titles. So the title should really convey a piece of knowledge, not simply say that you worked on this particular process, okay? So good titles communicate the main finding. Usually the title would be somewhat related, derived from that main finding sentence you wrote. might be different because you have to adapt it to title format, but basically don't write titles like functional characterization of gene X. That's not interesting. The only information in that title is that you work on protein X. That's all. We don't know more about it. The reader is not learning anything. But if you say protein X contribute to this process by doing this naughty thing to potato, then all of a sudden the reader now is learning something, is learning that protein X actually does this, and they are actually benefiting from your title. They have a better idea about what you're doing. And at the same time, they're also learning that you actually have characterized protein X. So that's implied in that title anyways. So really spend time working on the title. Maybe one way to make a great title is to make a list of the words that absolutely should be in the title and start making sure they are there. But the title essentially is really important. Think about again as your billboard. So do think hard about your title. On the other hand, I'm less of a fan of the abstract, or at least I don't think it's something you should write earlier, where you have a storyline, which is in a way a form of an abstract. But I think when you're done, when you have your whole paper done, it's worth it to go through this exercise of basically going through the text and highlighting those key sentences. The key sentences we talked about earlier, the ones in the storyline and so on. Go through the text and highlight the answers to the question, why did I do this? What did I find? And all that. And this is a useful exercise because if you can find those sentences, this exercise should be very easy because those sentences should jump at you. They should be so obvious in your text that you should easily be able to highlight them. If you can't find them or if they're like buried deep in the paper, then you have a problem. Then you're really in deep doo-doo because that means your paper is not well written. The key messages of your paper are not there, are not evident, and you really have to kind of revise it and edit it to make those sentences come out more clearly. So that exercise is very useful, and then you can copy paste those sentences and then revise them, organize them into an abstract. It's okay if the abstract contains sentences that are similar to sentences in the same text, maybe even identical. There is no rule against that. That's actually better because you don't want to have an abstract that doesn't really match the main text, right? So it's okay to copy paste and start with that. Okay, so we're done now. Now you can publish. Now you can post your paper in bioRxiv. We've done it. So you wrote your whole paper. This is step number 10, the last step. And I strongly recommend that at this step, you post your paper as a preprint. And I don't have time here to discuss that in detail, but main reason is that when you post it, when you finish your paper, it's all approved by all your co-authors and all that, and you post it as a preprint, then the whole community will be able to read it immediately. You'll be immediately sharing your paper to the whole community. It's a time for celebration. Everybody will read it. There will be time to go through a journal submission and all that. That can take a long, painful months or years, some cases. But at least when you share it as a preprint, you will be able to immediately enjoy the moment and immediately get feedback on it. And then it will also open your paper to peer review. Anyone can give you comments. And I can tell you from experience that we had quite a few cases where people send us some very useful feedback on papers that we posted as preprints. And I think certainly this is the trend, as you can see here. This is the trend with preprints and bioRxiv and what is now at about 2,500 preprint a month. And so definitely, especially for the students in the audience, then this is it's not just the future, it's the present, but it's certainly not going to go away. So maybe get used with the concept of preprinting earlier than later. Okay, so that's pretty much all I had, but there's one other benefit of really posting your paper as a preprint is that you can celebrate at that moment. When you finish the paper, you now share it with everyone and it gives you an opportunity to celebrate. It doesn't prevent you from celebrating again when the paper is published, but well, then you get 2 celebrations. How about that? That's pretty good. Okay, so that's all I had, and I think I think it's probably worth it to take a break because we've gone for over an hour. So how about we take a 5-minute break and then we open it to questions? Does that make sense? Yeah. Okay. So let me see. It's 5:15. Let's start at 3:20, and we'll have a discussion at that point. Okay. Okay, so now what I will do is I will take you fairly quickly through some bonus material. These are some writing tips. So hashtag pro tips. Okay, so read, read, and read. So this is about how good writing is really dependent on good reading. So as they say, one paper a day keeps the supervisor away. And I think one paper a day is probably a reasonable average a scientist can aim at. I think that's sort of worth to kind of consider the writing you do and relate it to that one paper a day. But I think really, in average, we probably should read or should manage to read one paper a day to keep us up to date on science. Why is this critical? Because we all learned in school that reading improves our writing skills, but also because our paper builds on previous literature and adding something to that literature. So you really have to have a perfect knowledge of the literature to be able to write your paper, in particular to write the discussion. And as we covered that earlier, especially to write the implications of your discussions and how your work relate to previous knowledge, but how also we move forward after that. Another point here is that good writing skills are learned and developed. So you really have to learn and develop your writing skills. Nobody is born a great writer. And so do your work on this, invest in this, get style book, consult on the internet. There's a lot of resources out there to improve your writing skills. So just do it, just invest in that. It's an important part of your job. Frankly, when you become a PI, writing is probably the most important activity you would be doing. So essentially, as a scientist, as a group leader, as a PI, you'd be writing all kind of stuff. And so it's very important to start investing in that early. And this is something that is really that you develop over time slowly. It's not something that you say, okay, I'm gonna learn writing, and after a month you're a good writer. It's really something that would take years and years to be good at. Read the literature critically. Some exercise I really like to do is when I read something and I really like it, I think, wow, this is very nicely written. I actually highlight it, I read it again, I try to dissect it. I even copy and paste that into a special folder I have for beautiful writing. And then I have my collection of things I read that I found are really very nicely written. And that applies to science, but that also applies to other things you might read, like quality writing, like the New York Times is very famous for having a very consistent editorial line. They have their own style guide and style book and so on. So maybe develop a process where you're regularly reading things like the New York Times or high quality writing, basically. Journalistic writing, good journalistic writing is very similar to actually science writing. It's the closest we can get to that. It's really about telling the story briefly, clearly, without any ambiguity, without hiding the key message, etc. So one advice I have, especially for those who are not native speakers, is read one New York Times article a day. I mean, this is how I developed my writing skills. I started when I was a student a long time ago, and I read the New York Times regularly. And that allowed me to really develop a sense of what is good writing, what is well written, etc. And there are some really beautiful sentences in the New York Times. Some of them I posted here in the Times font, of course. And this is so this is actually something I still do occasionally, is if there's you're wondering like, okay, how do is it centered on or centered around? So then you use the search button, the search function in the New York Times. You just search with the word centered, and then all of a sudden you'll have 10 examples of the sentences like this one, really beautifully written sentences with centered. Then you realize, oh, they always use centered on. Or never heard of him. He's arguably one of the smartest salary cap executives in football. I think I read this sentence like 20 years ago, and then it became Phytophthora infestans is arguably one of the most important pathogens or something like that. So that's kind of like how I develop my writing skills. Do you say whether or not, or do you say whether to, and stuff like that. So I just use the New York Times. It's very sharp writing, and I have to say that I find British writing to be a little bit more tedious. I think American style writing is more closest to what we want to write in science, in sense that it's something that is understandable by pretty much the whole planet, and not too complex, let's say, in terms of the style of writing. Okay, some pro tips, hashtag pro tips. Clarity, your paper is not a mystery novel. Make sure to say who done it many times. Say it early in your paper, say it in your title. It's the exact opposite of a mystery novel where you have to get all the way to the last paragraph to figure out who did it. So this is the exact opposite. Less is more, don't forget that, less is more. So we want to really have the paper focused. It's not a data dump. Don't write papers that are bloated with all the data, all the experiments you've done. You want to make sure they fit in there. It's much better to have a sharper paper with the focus. We talked about structure. When you write, really, it's the easiest way is first outline, decide on the logical flow of thoughts, and then start writing. One tip that is very useful is to use the first sentence to introduce the paragraph. Don't leave the conclusion or the description of the topic of that paragraph to the end. This is a mistake I've done for years. And when I realized it, it was really eye-opening, because all of a sudden, it's so much easier to write a sentence, an opening sentence that describes the topic of that paragraph, and then spend the paragraph basically describing or discussing that topic, rather than keeping that sentence to the end and then sort of rambling through some text to get to that sentence. Another tip that's out there about writing is produce that first draft as fast as you can. Good writing is not actually good writing, it's good editing. So if you can generate actually the first draft fairly quickly, then you can now focus on editing, and that's a lot easier to do than to actually write in the first place. So make an outline, write as fast as you can, and then spend a lot of time on editing. And frankly, editing is something that can go on and on and on. I mean, we have paper where we got to version 30 or something, where there's all this recurrent editing that goes on, and that's fine. That's how it should be. That's really how you can improve your writing, by good editing. I don't think anyone writes a first version that is perfect. So that's the way to do it. This is examples from that tip about using the first sentence to introduce the paragraph. So an example from the results, I think I told you that to do this, we did that. Is an example to determine whether gene X is expressed during potato infection. We perform gene expression analysis using this method, for example. Discussion, why a question, right? Again, for the discussion, we covered that, but this is an example. So start the discussion paragraph with a question. It's a great way to actually start the discussion paragraph. Why does Phytophthora have 65 copies of this gene? And then you can spend the paragraph answering that question, discussing that topic. Or start with a sentence that has an unusual observation. Compared to other eukaryotes, Phytophthora has an unusual number of copies of gene X. But you see what I mean? That last example is a very good one. I think many people instinctively will keep that sentence to the end of the paragraph. But it's so much more powerful and so much useful to your readers to start the paragraph with that sentence, with that unusual observation. I think most of the writing, your instinct would be to save that sentence to the end. But it's so much better to start by saying, okay, this is an unusual thing, right? Look, why does Phytophthora have so many copies of the gene, whereas other eukaryotes have only few? And then bang, spend the paragraph discussing that. All right, I know from experience this is a little bit controversial, but I think it's important to quote unquote plagiarize yourself. And what I mean by that is use templates. I think it's absolutely okay to use templates. I mean, we are in the business of science, of communicating science. We are not doing creative writing. So we're not really judged on the creativity of our writing in a way. We are really trying to communicate our science as efficiently and as clearly as we can. And so clearly there would be cases where there will be recurrence. How many times can you describe a figure legend for Western blot and sort of things, right? It's not, the point here is not to actually write it in 10 different ways every time you write a paper and describe a Western blot. I think the point here is really to communicate it clearly and ambiguously, and also without without anything missing. So it's actually, in a way, once you have written a good figure legend, for example, for a certain type of experiment, you now have again your structure for that figure legend, and you have all the pieces that should be in there. So it's a lot easier to use templates as a starting point. So I do recommend that. I also do warn you about plagiarism. Plagiarism means copying and pasting someone else's writings without their approval or without declaring that you've done that. You're not supposed to do that, but you certainly should discuss within your lab about using templates as within the lab as an exercise, and then again making sure that you're doing it in the proper way. So it's something to consider. I think this should not be a topic that's taboo, but there is a tendency sometimes to confuse plagiarism in scientific writing with plagiarism in creative writing. And I think it's two different things. And I think that discussion got a bit sidelines at time. But let's sort of make sure that we're communicating our science in the practical and pragmatic way, an efficient way, and not be overly worried about plagiarism as it is interpreted in literature. Okay, we talked about this before, but produce a first draft as fast as you can and edit. So good writing is good editing. By reading a lot, you will be able to develop a second sense about detecting bad sentences, complicated sentences, and learn how to simplify them. And this comes from reading critically and reading good stuff like New York Times and analyzing good writing. Less is more. Again, simple rule of writing: if you can delete it, delete it. Okay, if you can delete it and the sentence still clear, then for sure remove it. Edit, and edit some more. Very important. 3 rules I don't mess with. One is results are always in the simple past tense. We express gene X in plant. And the tradition, I was told, goes back to the idea that when you're writing a paper, this is not published yet. So it's not part of the scientific discourse, part of the sort of the knowledge base. So that's why you're basically being a little bit careful by writing in the past tense, because you're describing the experiments you did, you're describing the things you found, and not really assuming that they're already part of the knowledge base of science. So that's why we say we express gene X in the plant, or you don't say gene X is expressed in this plant or something like that. That's not, that would be for the next paper. On the other hand, published knowledge, things that have already been published, they are part of the knowledge base. So they, in contrast, will be in the present tense. So like Phytophthora genome contains 65 copies of gene X, and then you provide a citation. Every time you have a statement like that, a factoid if you like, then you support it with the reference. And that's an important distinction to have. And frankly, there's no excuse why anyone shouldn't follow that. Your results in the past tense, and published knowledge in the present tense with a supporting citation. I think this is point 3 is also a good one. Avoid the passive voice. Passive voice is really horrible. I know that some of the writing workshops say that you should never write with we did this, we did that. I think you should. That's my view. We've always done it. never really suffered from it. I think it's much better than the passive voice. The reason why the passive voice is not appropriate, in my opinion, for scientific writing is because the passive voice is by definition ambiguous. We don't know what was expected. What does it mean? It was not expected that 65 bands hybridized to probe X. Who did not expect that? Is that based on another paper? Is that based on your opinion? Is that like so? So the passive voice, by definition, actually lawyer type writing, it's meant to be ambiguous. It's meant to provide some wiggle room for interpretation, and that's not what we want in scientific writing. We want to be clear and unambiguous, as unambiguous as possible. So therefore, it's better to say we did not expect 65 bands, rather than saying it was not expected. Okay, where to publish? Well, we covered this a little bit earlier. So as I wrote, I think the first question to have here is your work relevant to broad audience, or is it more appropriate for specialists? This is the first question you answer. And then based on that answer, then you can decide already on what type of journals you can consider. Consider fit, not just the prestige, because some journals, even the broad readership ones, tend to be somewhat specialized, and they will be more open to certain types of paper than others. Like genomics type paper may not be appropriate to every journal. Some journals like genomics papers, some don't. Do you like the journal? Do you like the editors? Do you like the people associated with that journal? And keep in mind that open access papers do get more widely read and cited. All right, and now this is not about writing. This is about what happens when you submit your paper to a journal. Well, one thing is, if you haven't submitted papers yet, then trust me, they will be rejected. So get used to it. Sadly, this is how it is in science. Our papers do get rejected a lot. And this is a fact of life. So we just have to get used to it and develop a thick skin, which is hard. I think it takes many, many years to develop that. And even after many years, still not easy. But keep in mind that rejections can be appealed. Reviewers are not the enemy. Well, there are some people, but most of the reviewers will really do a good job and give you some feedback. Even if it's wrong, they're still giving you feedback, and it's always useful to have feedback. And if you don't agree with it, it will make you think about why you don't agree with it, which is useful in itself. So if you address the reviews seriously and use logical arguments, some cases you may have to change things, that's perfectly fine. Then you have a chance for an appeal. But in any case, you should really always take the reviews very seriously, even if you're resubmitting to different journals. One of the things that irritates me a lot and make me lose complete respect for authors is when I get a paper that I already reviewed for another journals, or I see it out and they completely ignored the comments that were given. I think that's probably I think it should be considered scientific misconduct to some degree. And I hope there will be one day where all the reviews will be open, and then that becomes really irrelevant whether people do that or not. Because I think that's, if you're telling someone there is something wrong in this figure, you're really lacking control and so on, and they just ignore it and go on and publish it, there's an issue there. There's really an issue. And they're not even addressing it in the discussion or something. That really says something about the kind of ethics and the kind of the standards that those people are applying to their work. So please do address the comments of the reviewers. Sometimes, of course, in many cases, they will be irrelevant. There will be minor things, or they will be asking you extra things. But in many cases, it's worth at least revising your paper or addressing qualification and so on to address those comments. And it's been known that appeals have been reversed, so you do have a shot at that if you make a good point. And keep this in mind. What's the difference between a diplomat and editor? Well, a diplomat says yes when they mean maybe. They say maybe when they mean no. And a diplomat never says no. But an editor, they say no when they mean maybe. And they say maybe when they mean yes. And they never say yes. So if you get a maybe, then you should probably be happy. Okay, and then to finish, just pre-print it. And I think this is really an important point. Back to my point about step number 10 and posting your paper in bioRxiv. Well, you know, it is liberating to post your paper in bioRxiv, because it is frustrating to go through that whole process of finishing a paper, submitting it, and then nothing happens for weeks, sometimes months, and then you get like a bang, a letter telling you to do another year or two of work. It's really frustrating. And I understand from talking to many early career scientists that this is a big turnoff for an academic career. When you get through your first paper and you get hammered, you're like, do I want to go through my whole life, my whole career, going through this horrible process? But fortunately, these days, your generation has an advantage. You have preprints, which we didn't have. So it's liberating to actually write your paper, share it with everyone. And it does take the pressure off from basically that whole process, where in the past, imagine, you can get scooped, your paper may not actually even get published at all. It's a totally different ballgame these days, because now you finish a paper and you can just celebrate and share it with everyone and get feedback. And in many cases, there's a lot of positive vibes that come back through social media and so on, and through feedback from your colleagues. So do take advantage of that. It's actually an advantage that we now have. Preprints do relieve a lot of that pressure of publishing and a lot of the tension and stress that comes with sort of waiting for ages for your paper to be out there. Okay, so that's all I had. Happy writing, happy publishing. I look forward to reading your papers, although I'll probably just read the titles. But remember, read, read, read. Okay? All right. Thank you very much.
Thanks, Sophien.
Thank you very much.
Thanks a lot, Sophien.
Thank
you.
Thanks, Sophien.
Bye.
Bye.
Thank you, guys.
Thank you very much.
Thank you, Sophien.
Right. Thanks. Bye.
Bye-bye. Thanks.